Okay, so in this video we're going to review protein synthesis, and it's really important that you understand the term synthesis. Right, so synthesis is going to mean to make. So the process of protein synthesis is actually making proteins, and it does this in two steps. Right, the first step is going to be transcription. Right, and this is where DNA is converted into mRNA. Right, this process occurs in the nucleus. Oops. And the reason why it occurs in the nucleus is because it's still dealing with DNA, which means that the DNA is going to be in the nucleus, and that's where the process is going to occur, because that DNA is going to be too big to leave. And that's the reason why it's going to be converted into mRNA, so that messenger RNA can leave and then go and make proteins. Right? And then what happens in transcription is that once the DNA turns into mRNA, the mRNA leaves the nucleus and goes to the ribosome. Right? And the reason why it's going to the ribosome is because the ribosome's main job is to make proteins within the cell. Right? Now, the second step is going to be translation. Right? And translation is the process where that mRNA is going to be changed into an amino acid sequence. And then all of those amino acids linked up together, once they're linked up together, they're going to form that protein. Right? So this occurs in the ribosome. And the reason why we're discussing proteins is because of two very important things. One, proteins determine our traits. Oops. And then our DNA codes for those proteins, which is why we say that DNA is in charge of our genetic information, and is in charge of really every function and characteristic and trait that we have in our bodies, because they code for these proteins, and those proteins are what determines our traits. Right, so let's look at some examples of how transcription and translation work. Right, so with transcription, right, that is where DNA is changed into that mRNA strand, right, and then there is a base pair rule that goes along with this as well. And then so the way that I set it up is that I have DNA on one side, and then I have my arrow, and then have RNA on the other side, or mRNA. So if we had an A in DNA, it's going to bind to a U in mRNA. If we have a T in DNA, it's going to bind to an A in mRNA. If we have a G, it'll bind to a C, and if we have a C, it'll bind to a G. So remember, the reason why the A binds to the U is because there are no T's in RNA. And so that's how you tell the difference between whether or not it's replication, which is the copying of DNA, so you're going to have plenty of T's in there. And then transcription, which is the process of DNA turning into mRNA. So you're not going to have any T's in the strand that you are making. You're just going to have those U's. Right? So an example problem, let's just say for DNA, I had TACGCATA. And then so we'll set it up where mRNA is right here. And then so that T will bind to an A binds to U, C binds to G, G binds to C, C binds to G, A binds to U, T binds to A binds to U. And that's it for transcription. Now the next part is going to be translation, which again is going to be the process of that mRNA being changed into our proteins. So mRNA and remember those proteins are composed of those amino acid sequences. Alright, now in order to do this, you're going to need to do two things. You're going to need to divide the mRNA into codons. Alright, and then in order to do that, you need to realize that a codon is 3 nitrogen bases. And then the start codon, so where you actually start this process at, is known as, or is AUG. And a good way to remember that is that school starts in August, which is abbreviated AUG. And then the last thing that you're going to need is a codon chart. And this codon chart is going to turn, it helps you turn the codons into the amino acids. Right? And so an example of a codon chart is this one. Right? Or you can have this one. And in the next video, I'm going to show you how to read those. So with, let's see, let's go ahead and do, right, so if our mRNA sequence was AUG, GUC, CGA, and then UGA, right, our first step was to divide the mRNA into codons. So what I'm going to do is I'm going to start with our AUG, and I'm going to draw a box around those 3. Then I'll just keep doing that until I run out. Alright, then the next step is going to make sure that you use that codon chart, and that's going to give you your amino acid sequence. And if you use the codon chart, you find out that AUG stands for MET, GUC is going to be valine, which is VAL, CGA is going to be arginine, which is ARG, and then UGA is going to be the stop codon. And you're going to keep translating your mRNA until you reach one of these stop codons. And you write stop, and then you're finished. Now, what happens if there is a mutation in the DNA? All right, so we are going to go into the 3 types of mutations. And remember, a mutation is going to be any change in the DNA sequence. All right, so we have 3 kinds. We have substitution, where one base is substituted for another. Right. We have insertion, where one base is added. To the DNA sequence. And then we have deletion, where you have one base is removed from the sequence. Now, in real life, there may be more than one base. It may be 20 bases that are substituted or inserted or deleted. But we're just going to do with one base just to make it a little bit easier. But you may come across problems later on where multiple bases have been substituted or inserted or deleted in one section of the DNA. So let's look at some examples of substitution. All right, so let's say that our original strand, and I'm going to make that an O, is going to be TACCA. Right, and then with replication, a mistake was made, and so our new strand may have sorry about that, guys. Our new strand, instead of it being, instead of a T being there, it could have been switched to an A, and then we would have A, C, G, C, A. So just to recap, our original strand was TACGCA, and then our new strand would, instead of that T being there, it'd be AACGCA. And then so because this T was swapped out for an A, that would be substitution. All right, for insertion, all right, an example would be, let's see, we have our original strand, and we'll still stick with the TACGCA. But then our new strand, after the insertion takes place, and let's just say there was a T that was added in, right? So there's a T that was added into the new strand, which would be an insertion. And then deletion works the exact same way, except something is deleted. So original strand could be TACGCA, and then let's just say this C was going to be taken out. So then that leaves our new strand as TACGA. And then so because these types of mutations affect the DNA, it will also affect the RNA that's made, which could affect the amino acid sequence, which would affect the protein. Okay, and we'll do an example of that in the next video. But that is basically the rundown of protein synthesis and then the 3 types of mutations. And then I'll do a video on how to put all of this together in like one problem. And that'll be in the next one.